Presented by Caltech.
People might be surprised there's even biology research going on at Caltech, let alone neuroscience research. We're mostly known for rocket science through the Jet Propulsion Laboratory, for earthquake research, and for research into astrophysics, gravity waves. But actually there's a rich history of basic biology at Caltech that has evolved in parallel with our strength in physical science and quantitative science. And so what Caltech has to offer is the marriage between the biology of the brain and the mathematics of the brain. And so Caltech has a very important role to play in the engineering of new approaches to studying the brain. I'm David Anderson. I'm the Seymour Benzer Professor of Biology and Biological Engineering at Caltech. I'm an investigator of the Howard Hughes Medical Institute and director of the Chen Institute for Neuroscience at Caltech. The work in my laboratory is focused on trying to understand how the brain encodes internal states, hunger, for example, sadness, fear, or anger, how those states are expressed by an organism in a way that's visible to other organisms, and how those states affect other brain functions like decision making. Advances in our understanding of the brain circuits of emotion will help us to understand and treat emotional disorders. Advances in understanding how we learn and remember will help us to treat learning disabilities. As we start to learn about how the brain processes visual information and makes decisions, that information may help computer-based technologies that will help us navigate the world in a more sophisticated and safe way. If you ask me what research have I been involved in here that I couldn't have done or wouldn't have done if I'd been at another university? One is my long-standing collaboration with Pietro Perona, who's in electrical engineering, to develop new computerized ways of measuring animal behavior. And then there's my collaboration with Ralph Adolphs, who's a professor in humanities and social sciences, to try to explain the neuroscience of emotion. So to have those collaborations with people in completely different fields like that greatly enriches the science that I think about, because I've had the most fun here when I talk to people who initially have no idea what I'm talking about and where I have no idea what they're talking about. And when it gets to the point that we can actually understand each other and we're on the same wavelength, then I know that something exciting is really about to happen. Good evening, everyone. I'm David Anderson, and thank you for coming to my talk, the Watson Lecture tonight on the inner life of the brain. Emotions are internal states of the brain that impact our decisions and our everyday lives and our health, but their biological basis remains poorly understood. Now, the modern study of emotion dates back to Darwin, and Darwin pointed out the homology in the study in emotional expression across species, including animals and humans. But Darwin was also well aware of the relationship between emotions and mental illness, as shown here in this plate from his 1872 book on the expression of the emotions in man and animals. He realized or inferred that the relationship there was a relationship between the disordered outward appearance or behavior of a person with mental illness and a disorder of the underlying corresponding emotion system. Now, despite important strides over the last 100 years, medicine continues to struggle with categorizing and treating psychiatric disorders. There hasn't been a fundamentally new approved psychiatric medication in the last 50 years. Now, part of the reason for this slow progress reflects a persistence of this somewhat outdated view that psychiatric disorders are due to chemical imbalances in the brain, imbalances in the level of chemicals such as dopamine, norepinephrine, or serotonin. This view arises from the fact that the symptoms of some of these disorders can be mitigated by drugs like Prozac, which change the overall balance of brain chemicals like serotonin. The problem is that serotonin acts on many different brain functions, so changing its levels globally can produce serious side effects. These side effects that are bad enough that they cause many patients to discontinue the drugs even if they're helping them. In other words, treating depression with these drugs is like trying to change the engine oil in your car by opening a can and pouring it all over the engine. Some of it will go in to the right place, but a lot of it will go where it does more harm than good. Now, an emerging alternative view of psychiatric disorders is that they are disturbances of brain circuits. So this view treats the emotional brain more like a computer than like a bowl of chemical soup. It's not that the chemicals don't matter, but they need to be targeted to the correct circuits in order to avoid these side effects. But if we're going to develop such transformative treatments, we have to study and understand the neural circuitry that underlies emotion. And so the question is, how do we do this? Now, emotions can be studied in humans using brain scanning techniques, and those experiments have linked emotions such as fear to activity in the amygdala, and my colleague Ralph Adolphs has been involved in that sort of work. The problem is that these sorts of studies, just by their nature, produce correlational results. That is, they don't tell us whether the brain activity that we're seeing is the cause of the emotion or whether the emotion is causing the brain activity. In order to do that, we need to get inside the brain and manipulate the brain to figure out how those manipulations change brain function. And we can't do those experiments in humans because they're invasive, but we can do them in animals. And genetic model organisms like laboratory mice and fruit flies are providing powerful new tools now that allow us to be able to mark, map, and manipulate specific cell types in the tangled jungle of the brain. So we can see, visualize, and map particular cell types. We can activate these cell types to see whether turning them on is sufficient to trigger a behavior, in which case we say these neurons are sufficient in the context of the brain to produce the behavior. And we can ask whether killing or inactivating these neurons blocks the behavior. If it does, we can say that these neurons are necessary, again, in the context of the brain for this behavior. And so the hope is that through these different types of causal manipulations, we'll learn about the function of the brain, and this will lead to new insights and eventually down the road to new medicines, in the same way that understanding the causal relationship between viral function and the function of the immune system has led to insights that have made it possible to produce vaccines for COVID. But now if we're going to study the circuitry underlying emotions in animal models, we have to confront the question of whether animals even have emotions. And for many of us from casual observation, it's obvious that animals, particularly our pets or animals that look like us, have emotions. We can just tell by looking at their expressions on their face. That's really an anthropomorphism, and it makes it difficult to know whether animals that don't look like us, that aren't mammals, like octopi or fruit flies, have emotion states. Now, in the past, there have been basically two ways of dealing with this. One way was Darwin's way. Darwin simply assumed that emotions, since they evolved by natural selection, were present throughout the animal kingdom. And he wrote in his monograph that even insects express anger, terror, jealousy, and love by their stridulation. At the other extreme is another hero of mine, the ethologist Nico Tinbergen. Ethologists study animal behavior from the outside in, and he wrote in a very hard-nosed way that hunger, like anger, fear, and so forth, is a phenomenon that can be known only by introspection. When applied to another species, it's merely a guess about the possible nature of the animal's subjective state. And so between these two extreme views, we hope that neuroscience, which neither Tinbergen nor Darwin practiced, will serve as a bridge between psychology, which studies emotional feelings like fear and anxiety, and ethology, which just studies observable external animal behaviors like freezing or flight, to get at what is in the middle. So how do we get at what's in the middle? How do we measure emotion states in animals if we don't have an emotionometer? So my colleague Ralph Adolphs and I have been working towards a view that emotions should be defined and considered and studied experimentally as internal states of the brain and the body that coordinate take various inputs from the environment and coordinate a whole battery of responses to these stimuli. And these responses can be behavioral responses, endocrine responses, autonomic responses, and part of the response is the subjective feeling that we as humans perceive. Now, we don't know whether animals have subjective feelings or not. There's no objective way to determine that. But from this point of view, we're not defining emotions as subjective feelings. We're defining them as a type of state. So we don't have to worry about whether or not an animal has subjective feelings in order to study internal states in those models. In other words, to make an analogy, we're saying that emotion to vision as subjective feelings is to redness. In the last 50 years, neuroscience has learned an enormous amount about how the brain processes signals that come in through the retina into the brain and maps them without worrying about how our brains generate the subjective experience of seeing redness. In the same way, we can learn a lot about emotion circuits without worrying about how the subjective feeling of fear or anger is generated. And I would say that we're about 50 years behind the study of vision in our understanding of emotion circuits. So if we're going to focus on the internal state, what kind of questions do we want to ask about this state? The general type of question is whether we're asking whether there are general properties that are common to different emotion states, as opposed to specific readouts that may characterize one particular type of emotion. And some of these properties, which Ralph and I refer to as emotion primitives, include the fact that unlike reflex behaviors, emotions are scalable or graded. They operate along a kind of continuous dynamic range of intensity. They persist for an extended period of time after the stimulus is removed. They have a valence, they're positive and they're negative, and they have generalizability, and there are other features as well. But the important point is that if we can figure out ways to recognize and study these emotion primitives, we can ask how they're implemented by brains. And I don't just mean mammalian brains, I mean animal brains as well, and whether an understanding of the implementation of these emotion building blocks, if you will, can help us to understand the evolution of emotion as a neural control mechanism, because emotions as central states didn't just appear on the planet with humans, they evolved as Darwin understood by natural selection. So to give you an example of scalability, I love this movie of an invertebrate, in this case an octopus, and I'll just play it because it speaks for yourself. So what you're seeing here is that as the threat, which is in this case the diver, approaches and gets closer and closer to the octopus shifts its defensive strategy from camouflage, or crypsis as it's formally called, to jet propulsion and escape. And presumably as the predator or the threat gets imminently closer, the level of the response in the octopus increases, and at different thresholds, the point is different behaviors are generated. So that's an example of what we call scalability. Now, persistence is just what we think it is, unlike a reflex response that terminates as soon as the stimulus has come and gone. In the case of an emotional stimulus, like a rattlesnake, the response in terms of our subjective sense of fear, our heart rate, our sweaty palms, persists for an extended period of time. And we want to understand how that persistence is regulated in the brain. A prevailing view is that the persistence is chemical in nature. It's due to circulating hormones in the bloodstream like cortisol. but it's also possible that the persistence is electrical in nature. It's a property of the networks in the brain. And one of the things that I'll be talking about today is our efforts to distinguish these possibilities. So I'd like to give you now a series of vignettes or examples of how we study these properties in the context of different types of internal states and their associated behaviors in both mice and also in fruit flies, if there's time. And our studies in mice focus on a deep brain structure called the hypothalamus, which is well known for its role in controlling feeding and metabolism, but which also controls innate behaviors such as defensive behaviors. And the hypothalamus is broken up into different territories, sort of like different states in the United States. And then within these states, there are different counties. And I'm going to be talking about one such county in a state, a part of the hypothalamus called the ventromedial hypothalamic nucleus, VMH. And this dorsomedial DM part of the VMH is what I'm going to be talking about for the next few minutes. Now, this structure lies in a pathway that is known to process innate fear responses to predators. And I want to show you that it's about 4 synapses in and an equal number of synapses upstream of the output of the brain. So this really is deep in the inner part of the brain where sensory percepts are being transformed into motivation state, emotion state, and action. Now we can study this region of the brain in a mouse in a very specific way because of a gene that specifically marks the neurons in this region. And this gene is called SF1. You don't have to know what it stands for, but through the magic of mouse engineering technology, we can build lines of genetically modified mice in which the only cells in the brain that are labeled are the cells that express this particular gene. And that gives us the power to mark and map and manipulate specifically those cells to find out what they do and how they do it. And there's a particular type of manipulation called optogenetics that I'll be talking about extensively this evening. And optogenetics is a technique for activating neurons with light. It was developed by Carl Deisseroth at Boyden and Caltech's Viviana Gradinaru when she was a graduate student in Carl Deisseroth's lab. And it basically works by transplanting a gene from a single-celled alga called channel rhodopsin 2 into a particular type of neuron and conferring on that neuron, thereby the ability to be activated by blue light. So we can perform this experiment specifically in these neurons deep in the brain, and we shine light on them by inserting a fiber optic cable deep into the brain. And then we can watch what happens to the mouse when we flick this light on and off and literally turn the neurons on and off like a switch. I'm going to show you a movie of this here. And when the light comes on here, we're turning the, we're activating the neurons, and you can see that the mouse stopped grooming and is frozen. And if you didn't see the wiggling of the cable, you wouldn't know that he was freezing. And when we turn the light off, he stops freezing and goes about his business again. And freezing is a very characteristic defensive behavior, particularly among terrestrial organisms. Even flies show freezing when they experience a threat. Now, the interesting thing about these neurons is that they don't just turn on freezing when they activate them. They actually activate a range of different types of defensive behaviors according to the intensity with which you activate them. So, for example, if you very weakly activate the neurons, you get avoidance of the area where the neurons were activated. At intermediate levels, you get freezing, as I showed you, and at even higher levels of activation, you get undirected escape or flight. The animal runs around and tries to jump out of the cage. So, like the case of the octopus in the movie that I showed you, these neurons appear to control defensive behaviors in a scalable, threshold-dependent manner. Now, what about persistence? Using a technique that allows us not to activate these neurons in the brain, but actually to record their activity in bulk. We recently performed an experiment where we asked, how do these neurons react when we introduce a predator in the presence of a mouse, such as a rat? And I'm going to play a movie of this, and you see the rat being introduced in this fairly heavy-handed way. This was before we had fancy machines to do this. You see the level of activity in the neurons going up while the rat is there. But now watch this. We take the rat away, and yet the activity of the neurons remains elevated, way above the original baseline, all during the time that the mouse appears to be in this stretched attend vigilant posture. So that indicates that these neurons can be persistently activated by a predator threat. Now, the question is, are these neurons actually functionally involved in this type of persistent response? This is, after all, just a correlation. And to figure that out, Ling-Yun Lee in the laboratory, a postdoc, designed a quantitative experiment to look at persistent defensive responses in mice. So you start with a mouse in an arena and you wave a rat over it for about 15 seconds. And then you watch what the mouse does. And these red lines are simply the traces that the mouse makes when it runs around. Before the rat is there, the mouse runs all over the bottom of the cage. But after the rat has been presented, the animal sticks to the walls and hugs them and avoids the center of the arena and continues to do that even after the rat has been removed. And this behavior, which is called thigmotaxis, is a classic sign of anxiety in animals. Now, if we measure the activity of these hypothalamic neurons under these same circumstances, you can see the rat activates the neurons. And again, even after we remove the rat, the neuron activity declines very, very slowly. So we wanted to know whether this persistent activity of the neurons after the rat is gone is necessary for the persistent circling behavior after the rat is gone. And to do that, we used a different kind of optogenetic tool, an ion channel that inhibits neurons when we shine light on it. And we triggered the inhibition of these neurons after the rat had been removed. And when we did that, the behavior of the mouse switched back to this normal non-circling behavior. So what that indicates is that this persistent activity is actually necessary for this persistent anxiety behavior in response to the rat. So these and other experiments have provided evidence that satisfy a number of the criteria or features of emotion primitives that Ralph and I have postulated. It indicates that there is scalability in the behavioral response. There's persistence in the response and in the brain act. There's a valence. When the animals have these neurons activated, they avoid the place where that occurred, indicating that this brain region tells the rest of the brain something bad is happening. And we showed that this can generalize to a number of different contexts. So we've done all of this without making any reference to the specific type of behavior that the animal is showing, even though those behaviors, such as freezing, are readily seen in cases of fear in humans as well as other animals. In addition, we can show, and others have shown, that if you activate these neurons, you produce the increases in physiological changes associated with fear, such as increased stress hormones, heart rate, and pupillary dilation. So we would say that these neurons are capable of coordinating an internal defensive emotion state. And if you want to think of that as fear, as long as you realize that we're not attributing any subjective experience or feeling of fear to the mouse, that's fine with me. Now, how far can we generalize this? In experiments that we did on fruit flies, we asked whether the well-known ability of flies to jump away from a looming stimulus like a fly swatter or a hand moving overhead, which has been characterized beautifully by Michael Dickinson and his student Gwyneth Card, whether that was just a reflex or whether that might also involve some sort of scalable, persistent internal state. And to test that, we designed a gadget in which we could keep flies in an inescapable arena with a transparent cover and then wave an infrared transparent paddle across the top of the arena, which would cause the flies to start jumping around, and we could film them through this panel. And briefly, what we found is that as we increased the number of swipes of the panel, the velocity of the flies increased. That is, they started walking around faster and faster. In fact, at a certain point, they stopped walking and they started hopping like popcorn. So that indicates there's scalability in this response. Moreover, even after we stopped moving the panel, it took the flies quite some time to settle down. The popcorn took some time to stop popping, and then eventually they would run around a bit and then settle down. And with my Caltech colleague, Pietro Perona, we were able to make a mathematical model of this behavioral response. And importantly, what this model shows is that there is a direct link between the property of scalability, the ramping up of the increasing intensity of the response with more dollops, if you will, of the input, and the persistence, the slow decay. And the easiest way to think about this is to think that the fly has something in its brain that acts like a leaky bucket that gets filled a little bit every time the shadow passes over its head, and that leaks slowly out. And when you stop filling it, it takes some time for what's in the bucket to leak out, and that shows the persistence. And if you want to think of that leaky bucket as an analog of a scalable internal state that is fear-like, you're welcome to do that as well. So now what about the relationship between fear and aggression, the fight or flight response? We've all heard and learned in school about the fight or flight response. And the interesting thing is that fight or flight is typically portrayed as being controlled by chemicals in the bloodstream, released from a gland above your kidney called the adrenal gland. There's adrenaline, which makes your heart pump more strongly, and there's cortisol, which is a stress hormone. The problem is that these hormones are elevated during both fight and flight, and they don't tell you anything about how the decision is made by the animal about whether to fight or to flee. And that presumably has something to do with the brain. It's not a decision made by chemicals in the bloodstream. And so we've investigated that by using similar approaches to what I've told you to study aggression as opposed to fear. And the fascinating thing is that if you look in the same football-shaped structure called VMH, where we were studying the fear neurons in green, literally right next door to these neurons is another population of neurons shown in red, which are marked by expression of the estrogen receptor. And if we build genetically modified mice to express channel rhodopsin and optogenetically activate these neurons, when we do that in the presence of an intruder mouse, the white mouse that is non-aggressive, it's either a castrated male or a female, you watch what happens when we turn the light on. We get almost instantaneous attack of the intruder mouse by the resident that we're activating as long as the light is on. Now, I emphasize that we stop these fights before there's any physical damage to the animal, but that indicates that stimulating these neurons is sufficient to cause the animals to attack. Conversely, if we use the trick of inactivating these neurons, we can ask whether these neurons are required for a naturally occurring episode of attack. And we have a naturally occurring episode by taking a male that has been made more aggressive by social isolation. I'll mention something about that in just a minute, and introducing a subordinate male, shown here with a lighter coat color, into the cage. And under those circumstances, the resident of the cage, the home cage, will spontaneously attack the intruder. And so we wait until the attack is underway, and then we fire these neurons with a laser, but now what we're doing with the laser is shutting these neurons down, not turning them on. So the fight starts, we get attack to the intruder, we turn on the light, and immediately the attack stops and the animal goes off into the corner. And I'll run that for you a couple of times. So that indicates that neurons are not only sufficient to trigger aggressive behavior when they're activated, but their activity is necessary, their continuing activity is necessary for ongoing aggression. Are these neurons actually active during aggressive behavior? To look at that, we use another technology which comes from, like optogenetics, aquatic organisms. In this case, the aquatic organism is a jellyfish, and jellyfish are fluorescent and green because they contain a protein called green fluorescent protein that makes them fluorescent. And you can engineer this protein and express it in neurons in the brain, specific types of neurons. And this form of the protein has been modified so that when a neuron is active and calcium comes into the cell, then the protein glows brighter. So you can actually see neurons being activated because they glow brighter when they fire. And so we've used technology developed by Mark Schnitzer, an optical physicist at Stanford, that allows us to literally mount a miniature microscope on the head of the mouse. It weighs about 2 grams, and through a kind of telescopic glass needle that is like a lens, we can actually image these neurons deep in the hypothalamus that are expressing this calcium-dependent jellyfish protein. And the inset here shows these neurons in a resident mouse, and you'll see we introduce an intruder, and immediately the neurons start to turn on and fire, and we can image the activity in the animal's brain while it's attacking or doing whatever it's doing to the intruder. Now, in order to make sense of this activity, we have to be able to correlate on a moment-to-moment basis the activity of different neurons with particular behaviors that are occurring. And in order to facilitate that process, again, working in collaboration with Pietro Perona and Kennedy, a former postdoc in my lab, and Christina Segelin, a postdoc in Pietro's lab, have used artificial intelligence tools to develop computer programs that can automatically segment and track the mice and tell us what behavior they are performing on an automated basis. And they can do this at the same time that we are imaging activity from the animal's brains. And so that allows us to synchronously stream both brain activity data and behavioral data from the video from the same animal at 30 frames per second. And we can use this then to precisely correlate changes in the activity of individual neurons with particular behaviors. And so, for example, what we found in this case is that there are neurons in VMHdl, these estrogen receptor neurons, whose activity goes up every time there is an episode of attack, and which is much lower when the animal is simply sniffing. And we can actually train a computer program to decode just from the pattern of neural activity when the animal is engaged in attack behavior rather than sniffing behavior. So these bars at the top are the actual observed behavior, and these bars underneath predicted attack are the predictions made by the computer programmer based on the underlying neural activity. And the assumption is that we, the experimenter, can tell by looking at the brain with a microscope whether the animal is in an aggressive state or not. The rest of the brain can do so as well. So together, what these studies indicate is that almost immediately adjacent to each other in this football-like structure, there are 2 subdivisions of the brain that are involved in controlling flight or freezing, and on the other end, fight behavior. And these 2 structures have remarkably similar functional properties. They both control their respective behaviors in a scalable manner. I showed you that for the fear neurons. The same thing turns out to be true for the aggression neurons as well. I didn't show you those data, but if we weakly activate those neurons, we cause the mice to sniff the intruder and to engage in dominance mounting, a weak form of aggression. And then as we dial up the intensity of the light, that mounting behavior switches to attack. So the behavior controlled by these neurons is also scalable. And then both of these neuron populations show persistent activity. I showed you the persistent responses of the fear neurons to a rat earlier. And it turns out that likewise in the aggression region of the brain, there are neurons that show persistent activity and that remain active at a high level as soon as you introduce an intruder into the brain. So these similar properties make us think that these neurons are doing similar things, except that they're doing them in the context of either fight or flight or aggression and fear. And we think that one way to think about them is that these neurons may be integrating input, combining input from a number of different sensory cues, and then converting that into some sort of scalable, continuously varying output. And then that output can activate different downstream structures to produce different behaviors at different thresholds. And that would be this scalability mechanism. Now, this is just a hypothesis right now, but it's a testable hypothesis. And consistent with this idea, both of these neurons, groups of neurons, send their projections to relatively similar regions of the brain, such as this trumpet-shaped structure here, which is called the periaqueductal gray, and then other structures in the amygdala as well. So both of these regions are broadcasting their information to large other regions of the brain, which is what you would expect for a brain structure that is controlling some kind of internal state, a central state that has to coordinate many different responses. Now, looking at this, brings me back to an issue related to emotions and mental illness. It's well accepted in the field that emotionality lies on a continuum from adaptive to maladaptive responses. And what I mean by that is that fear, for example, within a certain range can be healthy and adaptive. It's what stops you from getting hit by a bus when you step off the curb into the street. However, if the fear becomes too strong and stops you from going out of the house altogether and stops you from going to work or from buying food, then it becomes maladaptive and falls into the range of what a psychiatrist would identify as some type of a mental illness. And there can be maladaptations where the emotional response is too low as well. And reflecting this continuum of fear between adaptive and maladaptive levels, if one looks into the Diagnostic and Statistical Manual that lists different psychiatric disorders that are associated with fear, you see that there are many different disorders that are characterized that at their root reflect maladaptations of the fear system. Interestingly, if you do that for aggression, you find only one diagnostic category, which is called intermittent explosive disorder. This is basically somebody that loses their temper very easily. And even this one was only introduced into the DSM a few years ago. There are no other psychiatric disorders for which the primary symptom domain is aggression. Yet, every time there is a mass shooting and thousands of people are killed, it's pretty obvious to many people that there is something deeply mentally disturbed about the individuals that carry out that act. However, they cannot be diagnosed because there is no category syndrome to look up in the DSM whose main symptom is to carry out a mass shooting. And part of the reasons for this are political. There's a great reluctance on the part of the psychiatric profession to medicalize violence and that comes from pressure from the criminal justice system. If all violence is mental illness, no one will ever be able to be prosecuted and sent to jail for a crime. And there's also pressure from the mental health advocacy community. They don't want people to think that just because some people with mental illness carry out acts of violence means that all people with mental illness are potentially violent. It certainly does not mean that. The majority of people with mental illnesses are not violent, but it doesn't exclude the possibility that there may be specific disorders of aggression systems in the same way as there are specific disorders of the fear systems. And from the standpoint of a neuroscientist, given the functional and anatomical similarities between the fear and the aggression systems, it makes no sense to me for them to be treated differently in the context of mental illness. Now, I promised something about sex in my talk to get you all in here. And I do want to talk about one aspect of that, and that is what I call the paradox of sex and violence. As illustrated here in this beautiful diagram, it's well known throughout the animal kingdom that the presence of females enhances intermale aggression. Males most often fight with each other when they're competing over access to females for reproduction. So females make males more aggressive, yet the males almost never attack the females. They only attack other males. Why is that the case? To understand that, Tomomi Karigo, a postdoc in my lab, has recently gotten some insight into the neurons that control male sexual behavior in the mouse. And these neurons also live deep in the hypothalamus in a region called the medial preoptic area that is a few millimeters away from the area that controls the fight or flight responses. And it was possible for Tomomi to study the control of male mating behavior using male mice, not only by watching their mounting, but taking advantage of the fact that when male mice interact with a female, they sing to her. And they sing through ultrasonic vocalizations, which you can measure if you have the right kind of microphone in the cage. Importantly, when a male mounts another male, it does not sing, which is one of the reasons we think that this is a form of low-intensity aggression and not sexual mounting. But to make a long story short, Tomomi identified specific neurons in the medial preoptic area that promote mounting and singing. And not only can they promote it towards another mouse, they can even promote it towards a toy mouse, as seen over here. If you watch in the circle, you'll see the light come on, we're stimulating the laser, and the mouse crawls up onto the dummy mouse and tries to mount it and sniffs it. And down here, you can see its ultrasonic vocalizations. Now, not only can we promote mounting towards an inert, inanimate object, but activating these neurons in the context of aggression will actually switch aggression to singing and mounting. So here we have a naturally occurring fight where the intruder is attacking the resident, and we're going to turn on the light, and you will see almost instantaneously the resident switches to mounting and stops singing. So these are quite literally the make love, not war neurons. Now, the interesting thing that Tomomi found about these neurons is that not only do they promote a mating program, but they specifically send a connection to the aggression part of the brain that inhibits the activity of those neurons. And so when the animals are in the presence of a female and they're mounting, their whole attack system is shut down. And that can, we think, in part explain why males don't attack females, but rather only other males. And this is presumably due to control by specific female pheromones. But if that's the case, how do we explain the ability of females to make males more aggressive? Now, we don't know the answer for sure, but we have some interesting hints. So Da-Yu Lin, a former postdoc in my lab, identified an important input to the aggression neurons that comes from the amygdala and hypothalamus that seems to drive these aggression neurons in response to male cues. And Stefanos Stacharakis, a postdoc in my laboratory, showed that this specific synapse onto the aggression neurons is capable of showing an increase in synaptic strength when it is repeatedly used. It's a type of plasticity called long-term potentiation. And the more fighting experience the mice has, the stronger the synapse. Interestingly, Stefano showed that treating the mice with testosterone makes this learning effect on the synapse even stronger than it was in the absence of testosterone. So testosterone strengthens synaptic plasticity onto inputs that drive aggression. Now, it's well known in the behavior literature that the presence of females, particularly novel females, can increase testosterone in a mouse. And we've also shown that the presence of females can induce aggressiveness in otherwise non-aggressive mice. And so it's tempting to think that there might be a causal connection between these two that could explain how females are able to enhance aggressiveness in male mice, while at the same time the attack towards the females is inhibited. And that's something we want to explore in the future. Now, I'd just like to finish by telling you a story that has to do with the question of whether there are genes that control aggression that are conserved in evolution. After all, almost every sexually reproducing species on the planet shows aggression between males of the species and female aggression as well. And that raises the question of whether aggression was invented independently many, many times in evolution, or that it arose from a common ancestral form. And one way to look at that is to see if there are genes, that may play a conserved role in aggression. And we do that using the fruit fly Drosophila as a model organism. And Caltech's own Seymour Benzer, who was a physicist and molecular geneticist who became a behavioral neuroscientist, developed the fruit fly as a system for studying the relationship of genes to behavior. Seymour sadly passed away in 2007, but he showed that fruit flies could be used to study aggression and courtship and other social behaviors. And an interesting question that we've been trying to pursue is, are there genes that control these behaviors? And if so, do they control internal states that might be analogous to anger, or do the flies simply behave like robots? Now, many of you may not have even realized that fruit flies fight, but I can assure you that they do. This is an example of one male fruit fly attacking another male fruit fly using a behavior called lunging, where it rears up on its hind legs and snaps down at the male. And that looks even more impressive at 5,000 frames per second in this video taken by Martin Heisenberg in Germany, as it does in this rare video over here. And I call this the lucha libre or free fall type of Drosophila aggression. Flies don't fight they don't injure each other when they fight, as far as we can tell. They're mainly like sumo wrestlers trying to keep their opponent off of their territory, away from their females, and to stop them from eating their food. Now, using the genetics available in the fly, and again, automatic behavior monitoring technology that we developed with help from Pietro Perona, we discovered a group of neurons in the fly that specifically promote aggression when these neurons are activated. So these neurons appear to be analogous to the neurons in the mouse hypothalamus that we found, except that there are only 3 to 5 of these cells on either side of the brain. That is, these 3 to 5 cells are sufficient to activate aggression. And interestingly, these neurons are found in the male brain, but not in the female brain, which may account for some of the sexual dimorphisms. But where the genes come in is that these neurons, we showed, act in part by releasing a gene product, which is a small chemical messenger called a neuropeptide. And this is a molecule that is different from the chemicals like serotonin, dopamine, and norepinephrine and acetylcholine that you've heard about. These are slightly larger molecules, a short chain of amino acids, and they bind to a receptor on a downstream cell, and through that, they activate aggression. Now, why was this interesting? It became very interesting in the context of an environmental, well-known environmental influence on aggression, which is that of social isolation. So in flies, as in many other species, including humans, prolonged periods of social isolation make flies more aggressive. So here is aggressiveness in single flies that were housed and then brought together to fight, and almost no aggression in flies that are housed in groups in a vial. Interestingly, if we knock out the gene encoding this neuropeptide tachykinin, the single house flies become peaceful. And if we overexpress this protein in group house flies, they become aggressive. Moreover, we find, and this is work of a graduate student, Keding in the lab, that when the flies are placed in single housing and isolated for several days, the level of this peptide messenger RNA goes up. So we wondered whether a similar thing might be happening in mice, because this neuropeptide gene is present in mice as well as in humans. And mice, like flies, indeed are much more aggressive when they've been socially isolated for 2 weeks than when they're group housed. And remarkably, Muriel Zelikowski, a postdoc in the lab, found that when mice are socially isolated for 2 weeks, the level of this neuropeptide gene, tachykinin 2, goes up tremendously in the mouse's brain. If we label it with a green fluorescent protein from the jellyfish, the entire brain turns green in an isolated mouse. Now that's just a correlation, but we were able to show, using viral technology developed by Viviana Gradinaru and Ben Deverman at our Clover Institute here, is that if we overactivate the tachykinin gene and make the tachykinin neurons release more of their peptide in group housed mice, we can get these mice to be more aggressive as if they were socially isolated. So that indicates that the increase in tachykinin is really causal for promoting aggression enhanced by social isolation. Most importantly, when we tested a drug that blocks the receptor for tachykinin called osinostat, we found that that drug was able to completely reverse the effects of social isolation to enhance aggression. Furthermore, the drug also blocked the effects of social isolation to make the mice more fearful and more anxious, which are data that I'm not showing you here. And we only discovered this because we were led to it from the work that we did in flies. Now, in this period of COVID, there's been a lot of discussion about the adverse effects of social isolation on mental health. In fact, there was a lot of discussion about that in the press even before the COVID pandemic struck. But that, how social isolation affects our mental health, has really come to the forefront. Interestingly, the drug that we tested in mice that blocks the effect of social isolation has been tested in humans and has been shown to be safe, but it was abandoned in the clinic because it was never shown to be useful for anything. It was tested in schizophrenia and in panic disorder, and it failed, and it wound up on the junk heap of discarded pharmaceuticals that failed. But our new findings suggest that it might be worth re-examining this drug to see if it might be useful in treating the effects of social isolation. And unfortunately, the COVID pandemic provides a natural experiment in which it might be possible to investigate that. And we're currently talking with one of the pharmaceutical companies that makes one of the drugs in this family to see if they're interested. So that indicates then, in closing, how comparative studies of behavior and neural circuits in organisms that are widely separated in evolution by hundreds of millions of years, like flies and mammals, can be useful for identifying general principles of brain function and even identifying conserved molecules of function and behavior that could lead to new drugs for psychiatric disorder. And with that, I'd like to close and thank all of the talented students and postdocs in my lab over the years who've contributed to the work that I've shown. There are many more in the lab whose work I haven't had a chance to include for time's sake, but their work is just as exciting and interesting. And I'd especially like to thank my collaborators at Caltech, Pietro Perona and Ralph Adolphs, my co-author on our book on the neuroscience of emotion. Finally, I'd just like to mention that some of you who live in Pasadena may have seen a new building at the corner of Del Mar and Wilson Avenue. That's the new Chen Institute for Neuroscience. And we hope that in this beautiful new building, there will be many investigators who gather to try to understand how the brain creates internal representations of the internal world and converts that to perception, emotion, cognition, and behavior, and who will develop new technologies to solve those problems that may be applied to the treatment of brain disorders, not only psychiatric disorders, but also neurodegenerative disorders as well. So with that, I'll stop and thank you for your attention, and I'll be happy to take some questions.